start giving the test. We have very few time left for GPAT. So now without wasting much of time, I will quickly share the PPT. I would like you all to give your answer in the comment section so that we all can understand whether every one of you is how much we have prepared yet for the GPAT and how much yet is left to be prepared for the GPAT. So biochemistry is all about cycles, majority of the cycles, and the basics of biochemistry you have also covered in the 12th. So that generally does not become a problem. The problem comes when we are actually going to uh, see the cycles, and that is sometimes becoming difficult to remember maybe or retain them. And for that matter, the only thing or only shortcut is practice. If you practice these cycles, and you must have already done it during your preparation for sem 3 sem 4 maybe. So right now it's just a revision, but अगर तभी भी वो subject difficult था, तो only key advice is again just to revise the cycles. That will be enough, and to revise the basics. Norman, it is showing that recording is paused. I am just confirming, is that okay?
We are on live ना, so recording का need नहीं था इसलिए. If you want, I can.
Okay, okay. No, I am just telling you whatever is comfortable is fine.
Okay.
Okay, yeah. So I'll quickly share the presentation. Yeah. So yeah, so as I was saying, biochemistry is all about cycles that we deal with. So in carbohydrates, for example, see few things I am speaking which are not available on the slides. So I would request everyone to write it or note it down. Carbohydrates, we had studied many cycles. Now the important ones from the GPAT point of view, I am going to discuss today. Rather, I will be saying only the names because this is only this is not the forum where I will be discussing the entire cycle, but it's a forum where we will be discussing few important points, right? So cycles in detail, if you want, you can go back to my videos of revision of biochemistry cycles, where I have revised each and every cycle and enzyme in detail. As far as carbohydrates is considered, there are two parts. One is biosynthesis in every biomolecule. Now these biomolecules are all interlinked to each other. So when you study each and every biomolecule, so सबसे पहला question, what are these biomolecules? So they are nothing but compounds which are present in our body. Bio means that is related to our body or life, right? So these are molecules which are involved in our body for the different chemical reactions or different chemical processes that are required to make our body live. Say for example, right now if I am speaking, there are hundreds of different cycles going on into my body to allow me to speak, to allow my voice to modulate, right? So these is, we understand it most of the times pharmacologically, but what is the chemistry behind it? See, pharmacology always is a backhand of chemistry behind it. What exactly, what chemicals are, like we say in case of hormones, right, that these hormones are released. So similarly, what is actually the chemistry behind this, and that is what we are going to study. Now when it comes to carbohydrates, so the important cycles are glycolysis, which is the first cycle. Then we have HMP shunt, which is also known as pentose phosphate pathway or hexose monophosphate shunt. Then there is gluconeogenesis. There is Spory's cycle. There is glycogenolysis, that is breakdown of glycogen. Then there is glycogenesis, that is for making up or biosynthesis of glycogen. So in carbohydrates, majorly will deal around two molecules main. One is glucose and one is glycogen. And when we say about glycogen, it is nothing but a storehouse of glucose, or it is a polymer of glucose. We also see in carbohydrates the different classification of carbohydrates. Then when it comes to proteins, proteins are nothing but they are building blocks of amino acids. Uh, uh, right, rather, let me say it like that. Amino acids are building blocks of proteins, right? So these amino acids, how actually they are synthesized? What is the classification of these different amino acids? What are aromatic amino acids? What are aliphatic amino acids? Which contains hydroxyl group? That all is given in detail in the classification in the first page of any of your biochemistry textbooks, right? So classification is important. Another which is important in case of proteins is the cycles, that is synthesis of purines and pyrimidine. Synthesis of this amino acids, how actually it takes place, and where these amino acids are involved in the metabolism, right? Next, we come to vitamins. Again, vitamins may there are four different vitamins. Uh, uh, Rather, I will say there are two classification classes of vitamins. One is fat soluble and one is water soluble. In case of fat soluble, we have four vitamins A, D, E, and K. And in case of water soluble, we have two vitamins, vitamin B and vitamin C. Right. So now this vitamins, how they are synthesized. What if these then we have vitamin B complex is a very big vitamin. Now in this vitamin B complex, which exactly are the um, uh, B1, B2, what are they? B1, B2, what are the deficiency syndromes associated it? Then what if this vitamin is deficient? Can we take it outside from the diet? What are the diseases associated? So in vitamins, this part is important. And then last we come across nucleic acids, right? So nucleic acids, we also see in that the Chargaff's rule, that is DNA synthesis, then which is the basic molecule that we see. Then we see purines and pyrimidine bases, how they are synthesized, uh, how what is exactly how ATP is linked, what is AMP. So these are some important chunks of nucleic acids. Okay, then again one more part about it which is important is transcription translation. Okay, where little bit of biotechnology is highlighted. So let us start with the very first question, that is Cori's cycle. It is on Cori's cycle. Is gluconeogenesis from which? Now you all can start your using your comment box and give answers for this. Now, how do we solve or approach this question if it comes directly in GPAT? So सबसे पहले what should be the thought process? ऐसे ही अगर आंसर नहीं याद आ रहा है, how can you link things? Gluconeogenesis, you have to break genesis, some Latin terms. Now over the 4 years you will be knowing, uh, or you must have revised little bit, right? So that I am assuming some Latin terms you can break down and understand. Genesis is formation, glucose is glucose, and neo means something. Gluconeogenesis is formation of glucose from a non carbohydrate moiety. Okay, so here from a non-carbohydrate, when I say Cori cycle will not be from glucose. So one option is directly eliminated, right? So now we have glycerol, lactate, and propionate. Now, if you are confused, again you can see I am making you explain by the elimination law. Now, again, if you are say okay, I can see already few answers in the comment box. Confused that what to choose. So Propionate generally is not a starting material for any of the cycles, or we have not come across. So now what is left? Lactate and glycerol. So we have 50% options now eliminated. Okay. And the correct answer for this particular is lactate. Okay. So that is how you can eliminate the option and think of glycerol again. Why not? Because it's an alcohol containing moiety. And if you have to think in back ways, how can you synthesize glucose from an alcohol? Is it possible? No. So no, right? So the answer here is lactate. So this is Cori's cycle, which I have just put a snapshot. Okay. What happens exactly? Glucose in muscle is converted to glucose 6 phosphate, which is further by the cycle converted to pyruvate. Now, pyruvate is divided into two. One, it will form alanine because of transamination reaction, and other, it will be converted to lactate. This lactate will move to the bloodstream, and from there it will move to the liver. Once it is in liver, here there will be production of glucose that will take place. So actual production of glucose takes place in liver from lactate moiety. Lactate will be converted back to pyruvate. Pyruvate will be converted to glucose 6 phosphate. Now this will be converted to glucose, right? So this happens in liver. Next question. Sucrose is made up of dash. So let me see if we have any answers. Again, this is a direct type of question. The elimination here is kind of difficult. Okay. Still, if we see at the structures or if we see at the options, all the option contains glucose and fructose. So we know for sure that there is glucose and fructose that is going to make up sucrose. Now this is a difficult question, because if you see, they have not used any other sugar except glucose and fructose. Only what is difference is the conformational isomerism. So for that, you should remember one thing. Whenever we start with carbohydrates, I always say most abundantly used sugar is beta D glucose. Okay. Only in some places alpha D glucose is used. But again, if you see here, L-glucose के options we can eliminate, thinking that because most abundantly used is D-glucose whenever it comes into formation of a disaccharide. Then, so we have D-glucose and alpha D-glucose in two options, and then we have beta D-fructose in one option, and in the second option we have alpha D-fructose. So again, it's a confusion. So any one of you who can help me with the answer directly, or can we go to the answer? Any one of you can try. I can see only Isha responding. Every one of you, please respond. She says answer is A. Correct. Answer is A. Alpha D glucose and beta D fructose. So again, if we go by the logic that we had used, you can again get this answer as correct, right? Now there are different sugars. I have given here a few examples. Maltose is formed from glucose and glucose, and there is alpha 1,4 linkage associated. Lactose is formed from glucose and galactose. Trehalose is formed again from, it's a bisaccharide of glucose, so it is formed from two molecules of glucose. But here the linkage which is involved is 1,1 linkage, alpha 1,1 linkage. Next question. What is the starting material for biosynthesis of purine? Again, this is a direct type of question. Purines and pyrimidines biosynthesis is very important in case of nucleic acids. So let me expect answers from you all. Any one of you. I will just open comment section from my end on YouTube so that I can check. whether I can see any answers there as well. Noman, are you there?
Yeah, ma'am.
Can I see comments on YouTube as well? Are
Uh,
there any comments?
no, ma'am. Currently, there are not.
Okay, fine.
Hmm.
Okay, thank you. So even I have opened it. So I am waiting for you guys to answer. Come on. Yes. See, we have 8 to 10 people in the meeting at least. So at least that people can answer. This is a revision class. At least try to answer, right? So yes, many of you have answered correctly. Pragnya, Nita, everyone has answered. And it is phosphoribosyl phosphate, which is PRPP. Very correct. Now, again, this is a direct type of question. There is not much explanation involved here. You can have a look at the biosynthesis of purines and pyrimidines. Which of the following enzymes is responsible for the conversion of pyruvate to lactate in aerobic conditions? So what, see, in case of enzymes, when it is asked, no, you understand the materials or reactants which are involved. Now here it is pyruvate to lactate, so it's just kind of what kind of reaction? Is it decarboxylation or there is dehydrogenate? It is just a removal of a hydrogen that you should understand. If you know that, you can easily answer this question. Pyruvate to lactate. Let us look at the options. Well, first is pyruvate kinase. So when you use the word kinase, kinase is when there is formation of some enzyme. So this gets eliminated. Then there is pyruvate decarboxylase. So pyruvate decarboxylase means what? Again, this enzyme will be used when there is removal of CO2. Okay, so let us keep this aside. This can be an answer. Lactate dehydrogenase can also be an answer. And what is the fourth one? Alcohol dehydrogenase. So alcohol dehydrogenase, why? Because here we are nowhere, considering anywhere alcohol here. These are all acetate salts, right? So this, out of this, what is the answer? Now, if you know have an idea of the structure of pyruvate and lactate, you will understand that whenever there is conversion of this to this, there is nowhere loss of carbon atom. So if there is no loss of carbon atom, then there will be no decarboxylase coming into picture, right? So answer to this is option B, that is lactate dehydrogenase. Okay. So this is how your thought process has to work, and I would like you all to think in the same manner for the next questions that we are solving. Enzyme responsible for the phosphorylation of glucose to glucose 6-phosphate in the glycolytic pathway is dash. So glucose to glucose 6-phosphate, that means there is addition of phosphate group. Okay, addition of phosphate group can be by kinase. So if you see, all the enzymes are kinase here. Okay, now in this, if you see the most again, phosphofructo cannot be the answer because there is no fructose sugar that is formed in this case. So phosphofructokinase cannot be the answer if people are writing that. It is not when you convert glucose 6-phosphate to fructose 6-phosphate, that can be the answer, right? So answer is something related to kinase only. It is either hexokinase or glucokinase, because again pyruvate is not involved. So answer to this is yes, very correct. People have given the answer to me, which is A, hexokinase. Now in this again question, there are two options that are very similar, and that is hexokinase and glucokinase, right? So you have to think, picture out, understand, and then check. Because now our strategy in GPAT has to be solving as many questions as possible. Which of the following molecules is a direct product of the TCS cycle? Direct product of the TCS cycle. So citric acid cycle we've already studied in 12th as well. Now it starts with acetyl coenzyme A. So product पूछा है तो ये तो निकल जाएगा. What is left? NADH2 and FADH2. If you know the bioenergetics of the TCA cycle, we see that there are ATP molecules generated and NADPH and FADH2 molecules are generated. So at the for this particular question, answer is D. That is both B and C. I hope you all are understanding what I am trying to tell you all. Which vitamin is involved in the carboxylation reactions in the body? Carboxylation means what? Carbon dioxide addition. Okay, or maybe at times also removal. So these are no, generally it is sorry, it is only addition of carboxylic acid group. So which vitamin is involved in addition of carboxylic acid group or carboxylation reactions in the body? Now if you see here again, A, K, D and C. So C is a water soluble, all rest three are fat soluble vitamins. So vitamin A is retinol or retinoic acid or retinal. Then K is phylloquinone. Vitamin D calciferol, cholecalciferol. And C is ascorbic acid. So which is responsible in this case? Yes, answer to it is vitamin K. Again, this is a direct type of question. You have to think. This is where your concepts has to be strong. So then you will be able to solve. So all these biomolecules, are interlinked. So best way to study them is just draw all the cycles once, okay, or if you have already made your notes, just revise the cycles every day, so you will get the visuals. The more you see or more you visualize things, you don't have to रट्टा नहीं मारना है इसमें, structures draw नहीं करना है, okay, but understand करना है कि exactly क्या किस वजह से हो रहा है, which is again very clearly given in my videos earlier if you want to have a look. Which of the following is a coenzyme for transaminase enzymes? Now सबसे पहले तो coenzyme क्या होता है, कोई बता सकता है मुझे? Coenzyme, what do you understand by a coenzyme? So a coenzyme is nothing but it is going to accelerate the process of an enzyme. It will get attached to the enzyme for its activity. So generally it is due to some molecular or structural conformation. So when it gets attached, it is going to accelerate or direct the path of the enzyme to the particular substrate. Okay. Now your transaminase enzyme majorly which is involved is pyridoxal phosphate. Okay. Which of the following compounds is produced during the breakdown of purines? So we've seen purines and pyrimidine bases. They are part of nucleic acid. So what happens when there is a breakdown or metabolism of purines? What is the end product of metabolism of purines? And the answer to that is uric acid, right? So end product is uric acid. So if you see in certain people uric acid increases, that means somewhere the breakdown of the purines has increased or metabolism of purines has increased. In the pentose phosphate pathway, which is HMG shunt or PPP, which is also known as uh, hexose monophosphate pathway. Okay, the enzyme glucose 6 phosphate dehydrogenase catalyze the first step of oxidative phase. What is produced in this reaction? See, they have given the clue itself. Actually, pentose phosphate pathway यहाँ पे दिया है. Okay, and first step पूछा है. So there is something. See, there is only one pentose sugar that you can see here in this. So now can you all tell me the answer? I've given you the hint again. Any one of you? Pragna says A, ribulose 5-phosphate. Why not NADPH? Why not NADPH? Any one of you? For this, let me go and take you to that to the book itself. For this particular question, I would like you all to go through the book itself. I will just reshare the screen 1 second. Can you all have a look at the? Can you all see the book? Just tell me, can you all see the book?
No, ma'am.
Oh, I thought you all can see. I don't know why it is not getting shared. Just a second. Yes. So now if you have a look here, in case of HMP shunt, okay, we know that it's a pathway, it's a unique multifunctional pathway because there are many molecules formed and it is also known as pentose phosphate pathway. Now in the first step, that is oxidative phase, what is it? It is a glucose dependent NADP dependent enzyme that converts glucose 6 phosphate to 6 gluconolactone. Okay, like you can see here, this is the first phase of glucose cycle. Now here if you see pentose sugar, when it is formed very much at the end, that is in the second phase of the uh, cycle, which is non-oxidative phase. Here they are just saying, asking us about the oxidative phase. And in case of oxidative phase, what are the two things that you can see regulated are? What is formed is 6-phosphogluconolactone or 6-phosphogluconate, right? So now again, I will just reshare with you all the same slide which we were working on. And we will have a relook at the option. Is there anything called as gluconolactone in the slide? No, right? So what you can see here in the slide is NADPH, which is the oxy. And again, in the question itself, they have given you two hints and one they have particularly given this pentose phosphate pathway to confuse you. So again, in this type of questions, be careful. They have given a hint that it is a oxidative phase. So oxidative phase है तो there is something related to NADPH which will be formed. Now when I talk about NADPH, again here I would like to highlight what is it? It is nicotinamide adenine diphosphate hydrogenase, right? So in that case, niacinamide or nicotinamide, you can use these terms interchangeably. So these are nothing but vitamin B3, right? So again, this is how you have to study cycles, correlate and understand, and then you study. So from one question itself, You have so many meanings that can be made, right? Next, the substrate for the enzyme ATP synthase is substrate, you know, and so which is the substrate for this enzyme? That is what you have to find out, whether it is ADP, ADP, NADH or phosphoenol pyruvate. So again, this is a direct answer. ATP synthesize करना है तो कहाँ से करोगे? तो ADP होना चाहिए उसके लिए. So which is the substrate here? ADP. Okay. Next, which of the following amino acid is a precursor for the synthesis of serotonin? Serotonin is a hormone. This is again a direct type of question. So, serotonin can be synthesized from which of these amino acids? So, it is tryptophan. So, whenever you study metabolism of amino acids, that time in that cycles you can see this formation of these hormones, how they are prepared. Which of the following pathways is responsible for the production of NADPH? Which pathway is responsible? Whether it is glycolysis, pentose phosphate, beta oxidation, or gluconeogenesis. We have just now had a look, right? So it's a direct answer for this. That is pentose phosphate pathway. Which of the following is the end product of glycolysis? So what is glycolysis? It is breakdown of glucose, right? So what can be the end product? अभी ये तो सबको आना चाहिए. Come on. Yes. Yes. Correct. So answer to this is pyruvate. Very correct. Okay. Directly you can get these answers. In the urea cycle, which of the following compound combines with ammonia to form carbamoyl phosphate? Carbamoyl phosphate. So again, here which in urea cycle, which is the one which is involved is bicarbonate. Then which of the following processes occur in mitochondria? So again, remember mitochondria is generally for oxidative synthesis. So which of these can occur in mitochondria? Mitochondria is the powerhouse of the cell. So which of these can occur in exactly in the mitochondria? Any guesses? No one? Okay. So it is citric acid cycle. See, it is a citric acid is the one where you have to think, guys, where we are actually producing carbon dioxide at the end, right? So that is going to give out carbon dioxide rather. So where it is all this is taking place, it is in case in the mitochondria. Which of the following bacteria is commonly associated with peptic ulcers? Peptic ulcers. Which bacteria is associated with peptic ulcers? I can see answers already. Yes, very correct. Very correct. It is B. Helicobacter pylori. If you see, E. coli is generally associated with UTI, generally commonly associated with urinary tract infections. Then Streptococcus pneumonia is directly pneumonia. Staph aureus is again associated with bacterial infection and it can be varied. Okay, so peptic ulcer is something that is going to be responsible for hyperacidity somewhere. Okay, because of which because of hyperacidity it will reduce it will induce ulcers, right? So which bacteria is responsible for that? And that is Helicobacter pylori. Okay, so now we are going towards the we have come to an end somewhere of the biochemistry, and now we are going few towards the biotechnology side. Which of the following is a characteristic feature of gram positive bacteria? So is it outer membrane, peptidoglycan, lipopolysaccharide layer, or periplasmic? So microbiology, gram negative, gram positive staining, you must have done, right? So gram-negative bacteria example is E. coli, gram-positive Staph aureus, or Streptococcus. So yes, very correct. It is nothing but B, that is thick peptidoglycan layer. Then which of the following viruses is associated with the development of cervical cancer? See, now these type of questions, no, for biotech especially, if you are just reading the pharma headlines, no, day by day to day life में, तो भी ये कुछ answers कुछ हद तक you will be able to give. Okay, or microbiology, if you have to revise, just revise the synopsis of microbiology. That also should be enough. Oh, I have got many answers, and which is correct, which is HPV, that is human papilloma virus. Then which of the following is an example of a bacteria that forms endospores? Now this again you have done in your SY microbiology practical, right? So now which is an example of a bacteria that forms endospores? So answer is Bacillus anthracis. See, these questions are direct. I cannot explain much into it because what concepts you need to revise, that I will tell you all when I am explaining. Like say for example, I told you what is the principle of gram staining, you should know here, because that will help you to recollect the structure of the bacteria, especially gram positive and gram negative one. Which of the following organism causes malaria? This is the direct answer. We all know Plasmodium falciparum, right? Plasmodium vivax. Sorry, these terms are to be in italics. Which of the following is the causative agent of syphilis? Again, a direct answer. Chalo, last. Toxoplasma is responsible for toxoplasmosis. Then Enta-amoeba histologica is amoebiosis. Here it is similarly trachomitosis. You should know each one of them. Huh? What is the causative agent and why? You should know each one of them. Very correct answer. That is Treponema pallidum. Many of you have again given this answer. Which of the following is the most common cause of UTI? Oh, I said it just a few minutes before. Answer to this is very correct, E. coli. I'll just share with you the recent statistics. That is about 95%, more than 95% of UTIs is due to E. coli. Which of the following viruses is associated with cold sores? Again, direct answer is your herpes simplex virus type 1. Then which of the following viruses causes chicken pox? Smallpox, measles, Epstein-Barr, or varicella zoster? Answer to this is direct varicella zoster. Again, in this type of question, you can, although eliminate few, you can eliminate smallpox, measles. So बचता क्या है, Epstein-Barr or varicella zoster. Abhi, again, chicken pox is now the one which is almost eradicated from our country. Not eradicated, I will say, but it has lower down. Uh, uh, as compared to earlier, how we have vaccines now available for that also. So the virus which is responsible, polio is, I can say it is almost eradicated. So whenever now there is a polio case, or even if there is a TB case, the government takes a note of the case, and there is every possible thing that is done to, take measures that it should not be prevalent. Which of the following vaccines is made using a killed virus? Killed virus, measles, smallpox, polio, Salk vaccine, or polio Sabin vaccine? So answer to this is polio Sac vaccine. Now Sac and Sabin, what is the difference? That just have a look once. Just make a note and have a look ones. If I tell you right now, no, you will not take that pain. Thoda sa khud pain lo. Salk or Sabin, what is the difference? So that once you will see it, you will also remember it. Which of the following is the primary purpose of BCG vaccine? Why it is given? That is Bacillus calmette-guerin vaccine. Why it was given? Was it to prevent hepatitis, typhoid fever, influenza, or tuberculosis? Why this vaccine was given? It is the first vaccine which is given to children when they are born. Okay, and this is given specifically to prevent tuberculosis. Which of the following is a characteristic of live attenuated vaccines? Live attenuated means they are behosh karte basically unko, and they are inserted into the body so that antibodies are developed against that enzymes. Right now, again here it makes me aware that you also should know the structure of an antibody. That is why fork shaped structure of an antibody, what is the basic, what is the antigen binding site on the antibody. So all that features you should be aware of. So they are more likely to cause disease than killed vaccines, or they stimulate both humoral and cellular immunity. They are less effective than inactivated vaccines, or they require repeated booster doses. What is the correct answer? So answer to this is they stimulate both humoral as well as cellular immunity. Again, this concept of humoral, that is what is the difference between these two immunities? What is innate and adaptive immunity? What is the difference between them? ये particular जो terminologies है ना, that you should be familiar with. Okay, at final year or when you are approaching for GPAT, or maybe some third year students are also approaching for GPAT, you can qualify it by good marks just if your basics are clear. Which of the following is true about serum therapy? Serum therapy. It contains antibodies that provide immediate passive immunity, or it is used to prevent disease from exposure, stimulate the body immune response to produce immunity antibodies, and it is derived from bacterial cultures. So very correct. You have answered very well. That is it. It contains antibodies that is going to provide immediate passive immunity. That is nothing but about the serum therapy. And serum therapy is again one of the therapies which is mostly used in chemotherapeutic patients or cancer patients. Last question for today's session. Which of the following vaccines is contraindicated in pregnant women? Is it MMR, that is measles, mumps, rubella vaccine, diphtheria, hepatitis B vaccine, or influenza vaccine? Please try. I would request few of you to try. Isa, yes. I have got already few answers. Very correct. Answer is MMR, that is measles, mumps, and rubella vaccine. It is contraindicated in pregnant women. Why? Anyone can tell me what is the reason behind this? Why particularly this vaccine is been this is contraindicated in women? Anyone can tell me that? Anyone?
यस मिस
Yes.
क्योंकि इन्फेक्शन हो सकता है मतलब जो फीटस है वहां पे
हाँ.
आई एम नॉट श्योर बट मतलब स्पेसिफिकली जब हम वैक्सीन देते हैं ये सब तो जो
हाँ.
वुमन होती है उसको हाई रिस्क होता है कि मतलब बेबी को भी ये इन्फेक्शन मतलब स्प्रेड हो सकता है.
So then, dear, in that case, very good attempt. But again, in that case, why not these three? See, they have written in bracket inactivated in case of this, right? So why not these three will cause if these vaccines are given, they why they are not contraindicated, or rather how they will not Think. Think why they will not cause. So actually, the answer to this is these are live attenuated vaccines. Whether all these three are either given in dead form of living or inactivated, but these are given in live form. So if there is any chance the infection occurs, okay, behosh karke dete, that is what is live attenuated. So if infection occurs, then it can be a problem, right? So since because they are live attenuated vaccine, they are contraindicated in pregnant women. Okay. So here we come to an end for today's session on discussion of MCQs with respect to biochemistry and biotech. So biochemistry, at least I have tried to touch each and every topic, and biotech few. But I would request strongly on the points that I suggested to revise that particular points, that is in case of cycles, and in case of biotech, the basic structure of the staining technique, basic structure of the bacteria, and what we have missed out questions on is about basically on transcription and translation. So just a few rDNA especially, what is it and what is when there is a necessity of a transcription or translation, and what are post transcription and post translational modifications. So that is all from my end. Any doubts, or we can end up? Okay, so then I like to end up meeting here. Thank you all. Thank you very much.